Welcome to the Division of Education, Innovation and Energy e-learning program. I am Ms. Jamilla Bacchus, CAPE facilitator in the area of biology. Today, we are going to go into sexual reproduction in plants. This is showing you a little more information with respect to the structure of the anther. And you can see the shape of the anther. It's a four chamber, so you would have had 1, 2, 3, 4. What do you recognize here? You are seeing that this has already broken. So the 4 chambers have separated, and this line here, the line, what we call the line of dehiscence, the line where it would have broken, and you now have the pollen grains free to exit this anther. They simply stayed in there until they were developed, and when they were developed, the anther burst open and the pollen grains came out. What is happening inside of here? It's 2 processes taking place. You have meiosis and mitosis. Now, students, I know this is a topic you would have done in Module 2, and it is a topic that some persons, they breeze over and they don't put a lot of emphasis on. But you need to appreciate these topics, especially when we are doing embryogenesis, which is the formation of the, when we are doing the formation of gametes, gametogenesis. You need to appreciate this process, both gametogenesis and embryogenesis. We need to appreciate these types of cell division. Let's look at what is happening inside of the anther. What is happening inside of the anther is that initially you start off with a cell called the pollen mother cell. It is called the microspore mother cell. This cell, as you would recall, it's diploid, so it's 2N, right? Because all of the cells of the plant initially would have been diploid. They had 2 sets. Diploid means they had 2 sets of chromosome. One set of chromosome coming from the male parent and another set coming from the female parent. So every cell has 2N chromosomes, or they are diploid. So it started off with a cell inside of the, so inside of here, these cells in here, the mother cells, these would have been 2N, right? And what has to happen there, in order to maintain that diploid number, these cells have to go through the process of meiosis, which goes from 2N to N, from diploid to haploid, so that when the pollen grains, which is haploid, H-A-P-L-O-I-D, haploid, when this pollen grain meets with an ovule, which is the female gamete, we will maintain the number of 2N. We will get back the number of 2N. Good? This will be explained a little further on. So you have this cell. Remember, this is inside, this is happening inside of the anther. This diploid cell, which is the pollen mother cell, also known as the microspore mother cell. Remember, the pollen, let me write it here. The pollen grain is known as the micro, M-I-C-R-O, spore. And the ovule is known as the megaspore. The ovule is known as the megaspore. Okay, so you have a microspore in the anther, and you have the megaspore in the ovary. And what eventually has to take place is the microspore nucleus and the megaspore nucleus have to merge to form the new zygote. That is where we are going to go. But what has to happen here is that this diploid cell has to go through meiosis and produce cells. It produces cells. At the end of meiosis, it's going to produce 4 cells. And all 4 cells are going to be haploid. So you went from a diploid cell to a haploid cell. I wonder if you can tell me what is happening at this stage. This is meiosis takes place in 2 stages. You have meiosis 1 and meiosis 2. And at the end of meiosis 1, what is happening? Do you recall? At the end of meiosis 1, the homologous pairs separate. So each cell would have had half the amount they started from. So if we started with a cell that had 30, each cell would now have 15 already. But at the end of meiosis 1, you still have the chromosomes intact. You still have chromatids intact. Go back, please go back and recall meiosis. It's very important that you appreciate meiosis going forward. And now we have these 4 cells, each having half the amount of chromosomes at what we started off from. And these now will eventually give rise to our pollen grain. So these 4 will simply separate. And so from every 1 cell that started off inside of the anther, we are going to produce 4 cells, each of them being haploid. These are the cells that eventually will leave the anther and land on the stigma. Okay? We can now go forward. So this is what is happening here. We have the formation of the pollen grain. As you can see, these cells are actually going through meiosis. You can see we separate into 2, but you have further separation of each cell. So you have 2 at the end of meiosis 1, and then at the end of meiosis 2, these 2 would have already separated. So what eventually we will get would be 4. I wonder if you're seeing this. You're seeing here, if you're seeing 4, you're seeing 1 nucleus here, 1 nucleus here, 1 here, 1 here. So this here is what we call the tetrad structure that we saw earlier. We saw the tetrad structure here. So this here, what I'm showing here is a diagram. But what I'm showing here would be a micrograph of what actually happened. Somebody would have looked under a microscope and actually seen these. So this is not just a diagram. This is actually seen in the pollen grains. Now from here, as we saw in this one, we saw that eventually they separate out into their own individual cells. Likewise, here we've seen that. But what happens next is an interesting thing. What happens is an interesting thing. The nucleus of this cell, of this pollen cell, actually goes through a process called mitosis. See why we needed to have the foreknowledge of meiosis and mitosis? Mitosis here, which is a division of the nucleus of a cell into 2 daughter nuclei, good, will ensure. So here, one of these, each of these cells here, use a different color, try red. Each of these cells will eventually become this. This here is a pollen grain. The pollen grain, it started off here with 1 nucleus. And it would have gone through the nucleus. Remember, mitosis is the division of the nucleus of a cell to produce 2 daughter nuclei. I am stressing on that because, sadly, in some quarters, people are still saying mitosis is the division of a cell to produce two daughter cells. There are times when the cell does not divide into two. It remains with one cell. But the nucleus divides to produce two daughter nuclei. And once that nucleus has divided, mitosis has taken place. So if we see here, this one cell with one nucleus inside of it went through the process of mitosis, and you now have two nuclei inside of it. So this one cell developed to form this thing. This is the pollen grain that is actually going to leave the anther and go out for pollination. The two nuclei, as you can see here, one is called the generative nucleus. You see it here, the generative nucleus. All right, and it does have a little membrane around it. And the other one is called a tube nucleus. The tube nucleus plays an important role in ensuring that the male gamete and the female gamete would merge eventually. Okay? So let's recap quickly here at this point. We are saying for the formation of the pollen grain, also called the microspore, we would have firstly inside of the anther the process of meiosis taking place. And meiosis eventually will produce 4 cells, each having N chromosomes, one of each set of chromosomes. These cells will then separate, and each cell is going to go through the process of mitosis. So you first had meiosis, and then you have mitosis. Remember the one with the T, mitosis, is the one where you don't have any variation. The one meiosis, you had variation with crossing over and so on. And at the end, you're going to produce a cell with 2 nuclei inside of the one cell. So that's why I said you don't necessarily have 2 separate cells at the end of mitosis. You still have one cell, but you have 2 nuclei inside of it. Let's go. Good. So this here, you please take your drawing paper. You are supposed to be able to draw the structure, label it, and annotate it. Look at the different parts. You're seeing several things here. I've seen this question in a past paper, and I know some people were stumped with this question. Good? Because, you know, they say, well, that's a small thing, man. CXC wouldn't ask us that. Yes, they will. Good? So this is, remember, it is a cell. All cells contain cytoplasm. They contain a nucleus and many other structures. So the main things you have here, you have the cytoplasm, this jelly-like substance in which everything is sitting. You also have the nucleus. Now this nucleus here is the tube nucleus, and we'll see how this comes into play in a little bit. You also have a structure called the exine, and then another one called the intine. These are the 2 outermost layers, and they play an important role. We'll see in a little bit. This here is the generative cell, and this cell plays another important role in producing the gametes. Good? This area here, you see the word aperture here. It's also called a pit. And it is simply the location where you may have actually the growing out of the pollen tube. It's like a break in the normal tough wall that will allow some of the cell membrane to come in there to be able to form the pollen tube. Okay? Take a look at it. Understand how to draw this, be able to draw it, label it, and annotate it. Remember, an annotation is an explanation of a label. So for example, we can put with the tube cell nucleus, we could have annotated this by simply putting a dash like that. Of course, our labeling, we know from experience you don't label exactly like this. You don't have a capital T and a common, U. You should either have totally capital letters, small capitals, or script lettering. But this was just a diagram. But for your drawing, please do not use that as an example. But your annotation, so if you have a tube cell nucleus, you can have it's responsible. So under your annotation, you can either say something about structure or function. So you can say it's responsible for guiding, oh Lord, guiding the formation of the pollen tube. Excuse my handwriting, but it's getting better. It is responsible for guiding the formation of the pollen tube. So this nucleus, it directs the pollen tube down through the style. Okay? Question. Can you think of 2 roles that the exine plays in the life of a pollen grain? And as I said here, always think about structure and function. Always think about structure and function. So any topic you're on, think about structure. You're dealing with the kidney, think about what is the structure of the kidney. What is the function of the kidney? Because there are times they will ask you to justify why the structure is as it is. What about the function of this part that has to allow it to work efficiently? Good? So, can you think of 2 roles that the exine plays? Remember the exine? We saw the exine here. Right? The exine is this outer layer. It's a tough layer because remember this pollen grain has to fly through the air. The air is dry. The atmosphere is dry. The atmosphere has a lot of pathogen. The atmosphere has spores. So if that exine layer is not well structured, then the pollen grain may dry out and it will die. So the exine is very important in ensuring that the pollen grain survive. Right? I have the answer here. You can take a copy of it. You can take a picture of it, or you can come back and look at it afterwards. Because I know you're going to come back to this video a little later on to just get a second taste of it. Good? The pollen grain is made up of 2 outermost layers, the exine and the intine. And the exine constitute the hard outer layer, which is made of sporopollenin, which is the most resistant material to be known. And the intine is a thin inner layer of the pollen grain, which is made up of cellulose and pectin. So the sporopollenin is that tough exterior material which ensures the viability of the pollen grain. Right? It is in direct contact with the elements of the environment. It has to be tough and must also help to prevent the drying out of the delicate microscope. So that is one important function. It has to prevent the drying out of this microspore. But another role it also plays is that it helps to create a pattern, as we can see here. Each pollen grain from each species actually has its own pattern. And so when this pollen grain lands on the stigma, each species actually, there must be a certain amount of synergy. If the pollen grain pattern is off, it will not facilitate the pollination. It's that intricate. Good? So the exine helped to form this external pattern that would allow the pollen grain to accept it. While we were looking at the plant, the male part, we also need to recognize the female part of the flower. You have the ovary. So if you cut across, if you look at this, you actually see like a sweet pepper. Yeah, like if you cut a sweet pepper across, you will notice you have several structures. You have the ovule. Let me see if I get a different color. So this here is the ovule, which is the female gamete, right? Just as how the female in human beings have the egg or the secondary oocyte, this is the female gamete here inside of the ovary. You have this little stalk that holds it in place. It is called the funiculus. And then you have this middle area here, which is supplying it with water and nutrients. That is called the placenta. So again, the reason why I brought this out, you need to be able to draw this. If you look at your syllabus states that you must be able to draw a cross-section of a plant ovary and label and annotate the various parts. This here, of course, you can actually see this is a structure and micrograph, and you're actually seeing the particular cells that are present here. Good? What is happening in the ovary? You have similar to what was happening in the pollen, the anther with the formation of the pollen grain. Similarly, you have the structure occurring in the ovary where you have the formation of the female gamete, the female, what we call embryo sac. So inside of the ovary, again, you will have an embryo sac mother cell. Just as how we had the pollen grain mother cell, you had an embryo sac mother cell, and note, it is diploid or 2N, right? And the first thing that happens is that this cell goes through the process of meiosis. And so from one cell, remember at the end of meiosis, you're going to produce 4 cells that are all going to be genetically unique because they would have had crossing over and independent assortment occurring. Remember, go back to your meiosis. If you did not understand meiosis as we go along and we speak these terminologies, you will be confused. Good? So you need to go back and review meiosis and make sure you understand it very well. Now, when you have these 4 cells, what happens next is quite interesting. Only one will continue on. 3 of them will actually disintegrate. The thing is, you don't know which 3. It's a whole random process. So one cell will continue on, and what will happen next, quite interestingly, this cell, it will grow, develop, and then it will begin to carry out mitosis. So mitosis, this cell here, that was produced through meiosis, so these are all N. This was 2N. These are all N. And this cell, so this is still N. So it has 1 nucleus in it. We use a different color. We use white. No, black maybe. So you notice you have 1 nucleus here inside of it, and that nucleus has N chromosomes. Again, N stands for the number of different chromosomes. So if it had 15 different chromosomes, N is equal to 15, right? 2N, that diploid cell, will be equal to 30. So this cell with N chromosomes goes through meiosis. This is the first meiosis it goes through. Note, not the first stage of meiosis here, because meiosis has 1 stage. But it goes through 1 meiosis, and so from this, it produced a cell with 2 nuclei. Seen that? You first went through meiosis. You produced 4 cells, each with a haploid nucleus. And then one of these cells begin to go through mitosis, producing 2 cells, each with a haploid nucleus. So this one is N, and this one is N. Then a second mitosis takes place here. From here to here, a next mitosis takes place, and so you now have 4, because each cell goes through meiosis and produces 2. Every time meiosis takes place, you produce 2. Anytime meiosis takes place, you produce 4. So this was the first mitosis, this is the second mitosis, and this is the third mitosis. Good? So from here to here, you go from 1 to 2, and then from 2 to 4, and then from 4 to 8. Good? From 8 now, what happens is that inside of this structure, you have the arrangement of these 8 cells in such a way that you have 2 of them in the middle, and eventually they merge to become 1. So this here is 2n, because remember you would have had n and n. You all get that? Remember, each nucleus is n. These 4 are all n. So if n was 15, each nucleus would have 15 chromosomes. What happens now when the 2n merges here, this nucleus, it's called the polar nuclei, that now has 2n. If it was 15 we were looking at, then that will be 30. And all these other cells would be n. So the 8 nuclei are now arranged where you have 3 to the top, 3 to the bottom, and 2 in the middle, but eventually it merges to 1. So eventually you have 7 structures. Again, let me go it over again just for a short while, just so that you can grasp it. So let me erase and go again. Thank you so much, students, for staying with the program and following through. I want you to continue your work. In spite of everything, we have to continue learning, because learning is for life. It's for your development as a person, right? Not just to pass an exam, but it's learning for life, right? So I'm just reiterating the point that you had the process, and you started off with the embryo sac with a pollen mother cell, an embryo sac mother cell, and this was 2n. And this went through the process of meiosis. So you had 1 meiosis, you had 1 process of meiosis, you then had this 1 cell, only 1 of them will continue on, and this carried out 1 mitosis, 2 mitosis, 3 mitosis. So you had 3 mitosis. Good. So in all, how many divisions we had? 4 divisions. 1 meiosis and 3 mitosis. And then you will also have here 1 fusion, because the 2 nuclei at this stage will fuse to form the polar nucleus. And so that is 1 fusion. And these are the activities that will take place towards forming the ovule. So the ovule is the last structure that you will have. It's called the embryo sac. Before the male gamete merges with it, this is the last structure that you will have. Okay? So this is what the ovule would look like before it is fertilized. You're seeing the structure, and the inside of here is the embryo sac, as you see here. The embryo sac is just this structure here, but you also have some other structures that are around it that are there to enhance it. But the embryo sac is the area inside of the ovule that is going to eventually allow fertilization to take place and be prepared for the process of the formation of a new plant. Good? So we come back to the past paper questions. Distinguish between self-pollination and cross-pollination. How do you know the difference between them? I can hear you shouting your answers. Self-pollination will only involve the passage of pollen grain from the anther of one flower onto the stigma of a flower, either of the same flower or another flower on the same plant of the same species. Whereas cross-pollination is the transfer of pollen grains from the anther of one flower onto the stigma of another flower on another plant of the same species. That was the question that was asked in a CXC past paper question. Mind you, this question may have been worth 2 marks. Good? So I trust that you would have been able to answer and get your 2 marks. And then they asked, some legumes are adapted for both self-pollination and cross-pollination. Suggest why this may be advantageous to the species. Why do you think? What is the advantage of being able to have self and cross-pollination? Well, there are times when you may not have a male around. There are times when you may not have another tree in the vicinity. You may be a lone plant in the area. Good? And so you can continue to produce new members of your species. Good? So it will benefit you in having cross-pollination because it will bring in more variety, but will also benefit you of having self-pollination because you will be able to survive and produce more members of your species even if you do not have other members around. And the second question, part A, 1, distinguish between the terms pollination and fertilization. Well, we didn't quite do fertilization just yet, but I still wanted you to appreciate that they can ask for the difference between them. And some persons do, have some challenges with differentiating between the both. As we said, pollination is simply the landing of the pollen grains on the stigma. It does not involve anything further than that. Pollination is simply the landing of the pollen grains on the stigma of a plant of the same species. Describe 2 reproductive strategies used by plants to promote cross-pollination. We spoke about several of them. Do you all remember? Monoecious, dioecious, where you have the male and the female parts on the same plant or on separate plants. Right? Separate plants will be desirable because it will encourage cross-pollination. You can also speak of heterostyle, where you have different structures and different locations. And so as the pollen grains stick onto a bee or a pollinator and they have to go past a structure, they may brush off the pollen grains. You can also look at dictogamy, where some parts of the plant will be developed and open before the other parts of the plant. You also have self-incompatibility, where the pollen grains, if it lands on the stigma of the same flower, it will not produce a pollen germ tube. There's one I did not mention there. You also have self-sterility. So there's self-incompatibility and there's self-sterility. Self-sterility, it will land on the pollen grain, but land on the stigma, but it will not have fertilization. right? It will be sterile to its own self. Good? So those are the mechanisms which encourage cross-pollination. Right? So another activity, I would like you to be able to draw the pollen grain. Practice, practice, practice. I tell my students all the time, practice drawing and labeling. So you take your time and you do a drawing for your SBAs and so on. This is important. You can also be asked to do a drawing, not a diagram, a drawing where you actually look at the structure and follow the structure using your pencil, very light pencil, not crossing drawing lines and so on, not crossing, your labeling lines and all the characteristics you will know of your drawing lines. You can practice drawing the anther. Right? This is a past paper question again. It asks to complete figure 4 to show the missing stages in the boxes labeled A and B. You can do this in your time, all right? After the program would have been finished, you can go back to it and complete it. This was actually a past paper question. It was literally asking you to go through the stages of the formation of the female gamete, the formation of the embryo sac. If you remembered that we started off with a cell that was diploid or 2N, and it would have gone through the process of meiosis, and you would have formed 4 cells, and only one of these cells would develop. So they're asking you, what should you draw in this box? Because the question is asking you to draw in the stages in A and B. So here, you should have done something like you do your drawing where you have one of the cells enlarged and the other 3 cells are going to be broken down, right? And so you will have one, from here, you will have mitosis taking place where you have 2. So you went from a cell with 1 to a cell with 2, and 3 of them are going to disintegrate. So you see what is happening here? These 2, what will happen here will be mitosis. So you have mitosis here. You have mitosis here. So we have 3 mitoses and 1 meiosis. Remember those. 1 meiosis followed by 3 mitosis, and then what you have here, you have the fusion of the nucleus. So the nuclei, the 2 nuclei that are in the middle, they eventually fuse and form the polar nuclei, and so eventually they ask you to draw what should be down here. What you should draw would be the structure having the 3 cells at the top, which are called the antipodal cells. You have the 1 cell in the middle, which is diploid now, and you have 3 down here, which are 2 synergids and 1 egg cell. So you have 7 in the, I know my drawing is not the best, but if I was sitting, I would do a better job of it. So these are what they ask you to draw in. I just put in these here, right? But they ask you to put in the different, what you should see in box A and B. Use an arrow labeled C to indicate on figure 4 where meiosis occurs during the development, right? So you had to do something like this, and you put in your C. You put your letter C. And that's answering the question. This was a past paper question, and I can tell you some students were not, they did not do a very good job, right? And simply because some students, they feel there's a lot of work to learn off the different drawings, but it's not. You just have to practice, 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 and you will get it. So, mango. We want our mangoes, we want our fruits. We need to have pollination occurring. We need to have our fruits and vegetables being preserved, our species being preserved. That's why we study biology. We need to be able to appreciate that for organisms to continue, for the earth to continue as it is, we need to continuously have reproduction, and in particular sexual reproduction continuing. So these are the specific objectives we started off with, and you can tell me whether we would have accomplished these objectives, right? So I want you to keep working on the psychomotor, the drawings, to ensure that you can do a good job. So I want to thank you very much for being part of the e-learning experience, this e-learning program. And I trust that you will continue to follow the other programs and, gain a lot more insights and be able to apply yourselves in the exam room. I thank you. Have a good day.